It's really unusual. So I'm so glad to have all of you with us today. We're going to go ahead and start with Dr. Alicia Schrott from the Department of Pharmacology, Toxicology, and Neuroscience. She's joining us from the LSU Health Science Center. So with that, I'm going to go ahead and turn it over to you. Thanks a lot.
Thank you. Good morning, everybody.
Good morning.
I'm going to share with you some of the work in my laboratory looking at the effects of prenatal drug exposure. And as I do that, I'm going to kind of weave my career trajectory through throughout my seminar to kind of show you how I've ended up at this area of research rather than kind of starting with my biography. Okay. So one of the questions that I often get as a drug addiction researcher is, why do some people become addicted to a substance or dependent upon a substance and other people don't? Why can some individuals drink alcohol and not develop alcoholism and other people do? And one of the puzzling areas of research in the field of addiction medicine is, why are we not all created equal when it comes to the issues of addiction? And so I've just listed here some of the factors that affect whether or not individuals have a proclivity to develop substance abuse. And in particular, my laboratory has been focused on looking at gender differences as well as the effect of developmental history. And in terms of developmental history, one of the questions that we focused on is whether or not exposure to a drug in utero has long-term consequences on addiction vulnerability later in life. And these are, this is a recent press release from some studies that were done in clinical situations, reporting now that about one baby is born in prescription drug withdrawal an hour in our country today. So this is data that came across about 2 months ago. Received a lot of publicity in the news media, and was a Journal of the American Medical Association look at prescription drug abuse and the long-term consequences. And so one of the questions that we've been asking in my laboratory is, what are the long-term effects of being exposed to a prescription drug like Oxycodone or Vicodin if you're using it yourself or if you're exposed to it in utero? So one of the ways that we look at this question is by asking whether or not there are sensitive periods during development when drugs or alcohol will have more or less of an effect on the developing organism. And at the bottom here, I have a graph of when various physical structures develop in the body. And you can see that there are some organ systems or some organs that have a very narrow time window of development, unlike the central nervous system, which develops all throughout gestation, as well as developing during the postnatal period till you're about 20 years of age, 25 years of age, and then it starts going downhill. And so one of the questions that we've asked in my laboratory is, are there sensitive periods in development when exposure to drugs would have more or less of a deleterious consequence? And how I got into this area of looking at developmental neuroscience, I'm going to start now kind of weaving my research history kind of through the seminar. So I did my undergraduate work at Lafayette College in Easton, Pennsylvania. This is a school that's pronounced Lafayette as opposed to Lafayette, and I get lots of people that are confused by the fact that Lafayette College is in Pennsylvania, not in Lafayette, Louisiana. While I was an undergraduate at Lafayette, I was a biology major with a psychology minor. And I did research with a professor in the psychology department who was interested in play behavior. And rats, like children, have a very synchronized set of play behaviors that develop in about the first month after they're born. And so I worked with him on a research project looking at how play behavior could influence later aggression in life. And so I spent a lot of my senior year as an undergraduate sitting in a dark room watching rats play and then eat cockroaches or crickets. And I got very interested in the concept of brain development and why behaviors emerge at different times during development. And some of this came from my interest as a female teenager who did a lot of babysitting and working in childcare situations, and really being fascinated by the fact that children's behavior emerges along a pathway that's pretty set. Children crawl, and then they walk, and then they develop language. And what is it about brain development that led to this programmed set of behaviors that were being developed? So I went on to do my Ph.D. work at the University of Connecticut, which is in the oft-forgotten northeast corner of Connecticut in Storrs, right near the Massachusetts border. And I did my Ph.D. actually in a field known as developmental psychobiology, or developmental neuroscience. And my research was part of a program project grant with some researchers at Harvard Medical School. And what we did was actually do research on a mouse model of dyslexia. And this was really my first experience, and really developed my interest in how do you model complicated human conditions. And so this is a small piece of a paper that we published where we used neuroanatomical similarities between the brains of individuals with dyslexia. This is a human brain, showing what are known as ectopic collections of neurons. These are neurons that have migrated to an improper location in the brain. And they're found in a high frequency in individuals with dyslexia, particularly in the left hemisphere, in regions that are involved in language processing. Well, it turns out they're also found in certain strains of mice, particularly mice that have an autoimmune condition that resembles the human condition systemic lupus. And so my dissertation work was focused on trying to understand how these ectopic collections of neurons, as well as the immune system abnormalities that we saw in these mice, influenced brain development and led to behavioral changes in the animals. Now, obviously, mice can't read. So when we're talking about looking at models, how do we model a human condition like dyslexia? Well, we did a battery of behavioral tests on the animals using behaviors that had kind of mouse correlates to what we would see with reading. In other words, kind of complicated cognitive processing. And we were able to relate some of the behavioral deficits in these animals to these ectopic collections of neurons, and some of them to their immune system abnormalities, the fact that they had an autoimmune condition which led to the increased production of certain cytokines. So this PhD work led me into my postdoctoral work at the University of Colorado School of Medicine in Denver, Colorado. And there I was part of a developmental psychobiology research group that was, again, interested in human development. But I worked in a laboratory that was known for behavioral immunology. And this is kind of an interesting field which is started to receive a lot more attention, particularly from clinicians. And this is the idea that, that illnesses, sicknesses, immune system changes can actually influence your behavior. And this is due to the production of neuroactive compounds by the immune system, particularly pro-inflammatory cytokines. These cytokines can get into the brain or can be produced by the brain and actually change behavior. So we were particularly interested in emotional behavior because one of the things that was associated with chronic inflammatory conditions like lupus, like multiple sclerosis, other just general illnesses like the flu, is that there was increased incidences of conditions like depression and anxiety. Well, again, we had to look at how do you model this in a rat. If we wanted to look at anxiety or depression or surprise or fear in a human, we have well-established ways of doing that. But in an animal, it's a lot harder. You can't just look at a rat and tell whether or not it's anxious. Now, I do think that I can look at a cat or a dog and kind of get a sense of their emotion, but anybody who's worked with rodents kind of has a sense that you're not really going to get a lot from looking at their facial expression. So again, we developed a sophisticated battery of behavioral tests as well as molecular biological correlates to try to get at why these cytokines that are being released in these inflammatory conditions could influence the brain. And in particular, we saw changes in measures of anxiety and depression. Interestingly enough, now in the field of lupus research and autoimmune diseases, it's now understood that the anxiety and depression are often preceding the kind of rheumatology clinical symptomology that you see, and that the brain may actually be a very sensitive detector of changes in inflammatory molecules. So I continued my interest in behavioral immunology when I went on to the University of Minnesota Medical School in Minneapolis. And there I was in the Department of Pharmacology. And there I combined my interest in developmental psychobiology, behavioral immunology, and now I added pharmacology kind of to the mix, looking at how drugs may in particular affect the development of the brain as well as the immune responses. And so the research that I did there was in the field of PharmacoNeurolmmunology, or PNI, and in particular in the area of developmental pharmacoNeurommunology. So the question that we were asking, and have kind of continued to ask in my laboratory, is whether or not drug use or withdrawal from drugs during development can change how the brain, endocrine system, and immune system develop. So in other words, all of these three systems, the immune system, the endocrine system, your reproductive hormones, and your hypothalamic pituitary axis stress hormones, can all influence each other. And we were asking whether or not drug use and withdrawal would have direct effects on any of these systems, or potentially indirect effects. And one of the things that we were able to kind of look at was also the cardiovascular system. So even though I was trained as a neuroscientist, my kind of delving into pharmacology and immunology has led me to kind of expand a little bit out of the area of neuroscience and look at some cardiovascular and immunological effects. We were particularly interested in how all of these changes might affect the health of an organism. And kind of going back to that picture that I showed in the beginning, what would happen to an infant who was exposed to a drug in utero and then went through withdrawal after the infant was born, particularly if that infant had a prolonged hospital stay? Would that infant have compromised health because of the drug exposure, or could the drug exposure and withdrawal compound any other problems that the infant may have in the hospital? So some of the work that I did at the University of Minnesota used a chicken embryo model, and I want to talk about this for just a second because it's kind of an unusual model for a biomedical researcher to use. But it turns out the chick embryo young chick model has some real advantages for doing especially developmental drug research. First is that you don't have a mother, and even though we are really interested in how the mother affects fetal outcome, sometimes the mother gets in the way. And so in this case, we can administer drugs directly to the developing embryo without having to worry about maternal confounds. You can use large numbers of subjects, so you can screen for drug toxicity relatively easily. And the chicken embryo and the young chick has been a really well-established model by developmental biologists, developmental neuroscientists. So there's a lot known about its development. And the embryo is actually very accessible. And I'll show you that in a second. Interestingly enough, the chicken embryo is precocious at hatching. In other words, its eyes are open, it can move around, it's fairly independent. This is in contrast to the rat, where the rat is born altricial in that its development is more like a human embryo. And so we can actually do assessments on the young animals. So how do we do this chicken model of prenatal drug exposure? Well, we incubate large numbers of eggs, and then through a process of candling, we can actually visualize the embryo through the eggshell. We can actually drill a small hole into the egg and then inject drug directly into the amniotic fluid, where it gets picked up by blood vessels and transported into the embryo. We then allowed the chicks to hatch and then looked at their behavior postnatally. Or we could extract the embryo and measure markers in the embryo just by opening up the egg. Or we can visualize what happens when we do withdrawal. So this is an example of one of our designs for one of our studies looking at the effects of opiate dependence. So in this study, we gave an opiate that's very similar to methadone. It's called LAM, noracetylmethadol. It's a very, very structurally similar to methadone. We can determine whether or not this Norlam compound is acutely toxic. We can induce withdrawal in the chicken embryos by injecting an antagonist, an opiate antagonist, in this case naloxone. So this would allow us to model what would happen to a woman who's pregnant. She's dependent upon an opiate, finds out she's pregnant, and then decides to stop taking the drug. Or she goes through a detoxification program. What happens to the embryo when it undergoes detoxification? Or we can just let those eggs hatch, and we can look at withdrawal postnatally and see what happens if the embryo is allowed to hatch and then undergoes a postnatal spontaneous withdrawal. And one of the things that we can look at, I'm just showing you one slide of data. This is embryonic data. Is one of the advantages of using a chicken embryo is that we can actually visualize the blood vessels. So this would be similar to doing an ultrasound in a human, but we could actually look at blood flow by measuring the diameter of blood vessels. So this is an animal that's a control. This is a blood vessel. This is just a suture that's our kind of our reference guideline for measurement. And then this is a blood vessel of an egg, of an embryo, of an egg where the embryo is undergoing opiate withdrawal. The embryo had been given a drug and then injected with naloxone. And what we saw was that there was dilation of the blood vessels during opiate withdrawal, and that we could block this by pre-treating the embryos. Well, we were somewhat limited in what we could do with the chick embryo model. So when I moved here to Shreveport, I embraced my inner pharmacologist and started pursuing in more detail developing model systems to study the effects of prenatal drug exposure. And in the past, much of the research had focused on looking at heroin and methadone treatment. Methadone was given to individuals who were dependent upon heroin. But more recently, we know that prescription drugs like Oxycodone and Vicodin, Oxycontin, have become more problematic. So I switched the opioids that I was looking at and started working on developing a model of prenatal oxycodone exposure. And this was really led by some interesting demographic changes that have occurred in the last 10 years, last 10 to 15 years, over who is using drugs and how they're using them. So traditionally, heroin users, and this is a graph where you have to do some gender bending. The males are in pink and the females are in blue. I'm not sure whether they did this intentionally or not. This is from the government agency. But when we look at heroin use, the traditional heroin user is a male. When we look at non-medical uses, and this is, I should preface this by saying, this is non-medical use of not legitimate use, but abuse of oxycodone, there's a much narrower gender gap. And actually some recent studies have shown that females are now using at equal or greater rates than males. And in general, oxycodone users are younger than heroin users. And this is data on who is abusing prescription drugs and what drugs individuals are using. This is looking at who started using a drug in the last year, if you were ages 12 to older. And the most common drug that people start using is marijuana. But the second most common drug is pain relievers, much higher than what you see with ecstasy, cocaine, LSD, heroin, or PCP. And there was one other piece of data that really kind of led us to look at this, was that particularly among adolescent women, if you were pregnant, you were more likely to be using an illicit drug than if you were not pregnant. And this is really interesting because if you look at the statistics after the age of 18, fewer pregnant women actually use drugs than non-pregnant women. But for young females, almost a quarter of them are using some kind of illicit substance. And there was data that suggested that there were long-term effects of prenatal opiate exposure on things like cognitive deficits, anxiety, aggression, alterations in stress responsiveness. And so we wanted to ask the question, if you were exposed to a drug in utero, are you more likely to develop abuse problems as an adult? And so I'm going to skip over this just to go to our rat model. How do we do this in a rat? What we do is we actually treat our animals for 28 days with oxycodone. So these rats, these female rats, are stably dependent. So this isn't a mom who all of a sudden she gets pregnant and then starts using oxycodone. This is modeling an individual who had been using oxycodone and then became pregnant. And we breed the rats for approximately 10 days or for 10 days, and the pregnancy is approximately 22 days in length. And then the pups are born. They undergo spontaneous withdrawal over the first 2 days of life, and then we do assessments on them as adults. So one thing that was important for us to look at initially was whether or not our drug was causing reproductive toxicity. In other words, would a female actually even get pregnant if she was, and carried to term if she was using oxycodone? So we developed a model where we did not see reproductive toxicity, where there were no differences in the amount of weight that the female rat gained during pregnancy or the number of bursts, a number of live bursts. So what we're looking at here is not just a toxicity effect, but an actual pharmacological effect. And we found that the pups do undergo a transient withdrawal after they're born. So the pups here are, the prenatal oxycodone treated pups are in the blue bars, and they have lower weight, especially across the first week of life. They do eventually catch up. And they do show a stress response. This is levels of corticosterone, the stress hormone in a rat. And you see that the animals undergoing withdrawal, particularly on day 1, are stressed. They have an increased stress response. So in order to see whether or not they developed abuse liability in adulthood, we looked at a task called conditioned place preference. This is a behavioral task that pairs a novel environment with exposure to drug on one side and a different novel environment with exposure to a vehicle on the other side. So on one side, their floor was had a barred floor, black walls, and we used a mint scent. And on the other side, there was a mesh floor, white walls, and a lemon scent. And on one side, the animals got oxycodone, and on the other side, the animals got water. And so we looked to see whether or not the prenatal exposure had any effect on where they chose to spend their time after receiving drug treatment. So first thing we had to make sure was that the prenatal oxycodone didn't affect their basal preference. Somehow it didn't make them hate the smell of mint or be really afraid of mesh floors. And there was no difference in their baseline preference for either males or females. But when we looked at males, this is their baseline performance. These are the animals that had water in utero. These are the animals that had oxycodone in utero. The animals that had oxycodone had a stronger place preference. They spent more time on the side where oxycodone was paired than on the other side, especially in comparison to those that were not exposed to oxycodone in utero. This suggests that the rewarding or reinforcing effects of the drug were stronger if you were exposed to the drug in utero. We went on to look at some anatomy and neuroanatomy to see whether or not, in addition to behavior, we saw neuroanatomical evidence of this. This is a region of the brain known as the nucleus accumbens. It's known as the reward center of the brain. It's part of the reward pathway that gets activated whenever we have anything that's rewarding: food, chocolate, sex, drugs. The nucleus accumbens lights up. And we use the immediate early gene CFOS as a marker for this. There are 2 regions of the nucleus accumbens, the core and the shell, and animals that were exposed to oxycodone show a greater activation in both the core and the shell than those that were exposed to water. So kind of in summary, these data suggest that prenatal oxycodone may alter the abuse liability in adulthood. And I didn't show you the data, but females were less sensitive to this. We had stronger effects in males. And we still need to determine whether or not it's the opiate exposure or the withdrawal that's driving this. But one of the questions that we have is whether or not there are changes in receptor populations that might underlie these changes. So, for example, opioids bind to a particular kind of opioid receptor, the mu-opioid receptor. The dopamine receptor is known to be involved in the rewarding properties. We know that stress reactivity may be involved, and we know that cognitive processes could also be involved. So this summer, the LBRN student, Adrian, who's working in my laboratory, is looking at the distribution of mu-opioid receptors. We'll also look at some dopamine receptors in tissue samples from animals that were exposed to oxycodone or water in utero and then given oxycodone in adulthood, so that we can determine whether or not these receptor changes or other molecules that we'll look at are involved in the changes that we see in the behavioral outcome. And I will end there, and if anyone has any questions, I would be happy to answer them.
I have a question.
Sure.
Whenever you were at Minnesota and you worked with the chicks,
yeah,
were the results that you got there the same with the rats you've gotten here?
That's a good question. We didn't quite ask the questions in the same way. We asked a couple of questions that were very similar. One, we looked at whether or not embryonic withdrawal or early withdrawal is more problematic. And this has also been supported in the human literature as well, is that it's much harder on the embryo to undergo withdrawal than it is for the neonate. So if you have to undergo withdrawal, it's better to do it postnatally than prenatally. We saw that in the chicken embryo. We see it in the rat, and it's also been seen in the human. We were also able to establish in the chicken embryo that if you have to go through withdrawal while you're pregnant, what would be equivalent to the second trimester is actually the safest of all the times to do it. That seems to be, we haven't been able to model that quite the same in the rat, but it does seem to be the same in the human. And a lot of that actually has to do with the changes in blood flow. One of the things that happens during pregnancy is the uterine blood vessels transport all the food to the fetus. They also take away all the waste. Those blood vessels can undergo vasoconstriction or vasodilation and vasospasm. And withdrawal has a lot of effects on blood flow. And so it looks like during the second trimester, during the third trimester, you get a lot of growth, and that's why it's problematic. You're not getting enough food in there. During the first trimester, it tends to interfere with reproductive hormones, and there tends to be a lot of miscarriage. But in the second trimester, it seems to be kind of the safest, if there is a safe time, to undergo detoxification. So we were also able to establish in the chicken embryo model that we then did in the rat model, the role of stress hormones. That there is a big release that an embryo can actually feel stress and release stress hormones when it undergoes withdrawal. That was easier for us to establish in the chicken embryo, but we've also been able to see it in the rat. Other questions? Okay.
Let's all thank our speaker for being with us.